In the last video, I very briefly introduced the different types of receptors and extracellular messengers that the cells use in order to communicate with each other. And I said I was going to talk about G protein coupled receptors in the next video. So as you can see here, I have drawn a G protein coupled receptor. And as you can see, there are 7 different alpha helices. So each one of these different parts is called a alpha helices. So they're in the alpha secondary conformation or secondary shape for a protein. So it is these ringed loops. There are thousands of different types of these G protein coupled receptors. So right down here. Thousands of types. We actually have 1,600 genes that transcribe G protein coupled receptors, and we only have 30,000 genes in our body. So that's an insane amount that are dedicated just to these G protein coupled receptors. Like I said, they always have 7 alpha helices. And they have 3 loops on the cytoplasmic face. So you can see these 3 loops are connecting these alpha helices. And then they have 3 loops on the cytoplasmic face. So the part that sticks out is variable, so that it depends on what type of ligand they may bind. And they're named G protein coupled receptors because they interact with what is called a heterotrimeric G protein. So a G protein is, there's 2 different types of classes, and I'm just going to kind of briefly introduce how the G protein works, and then in a next video I'm going to give an example of activating this G protein coupled receptor, which is right here, and then the associated G protein, how it becomes activated and how it may activate some downstream pathway. So a G protein coupled receptor, first of all, there are 2 different types. So there's 2 classes of these receptors. So they are GTP aces. So I'll write right here, GTP aces. And so when they have GTP attached to them, they are active. And when they have GDP attached to them, they are inactive. So GTP just stands for guanine triphosphate versus guanine diphosphate. So there are monomeric, and they're small, so examples of that would be RAN, SAR1, something like ARF, actually, ARF is one, ARF1. So these are ones that I've shown in previous videos, and I'll show them in further videos just to give a better understanding of these monomeric types. And then there are trimeric, which are the more common ones, which is actually what I've drawn up here, is a trimeric G protein. Trimeric. So they have 3 different subunits. So right here on the left is the alpha subunit, so alpha here. And then right in here is your beta subunit. So this is beta, and then this is gamma. So on the alpha subunit, there is a binding portion for your GTP or GDP, which I will explain in another video, just how the binding of the GTP will cause a conformational change in this molecule here. But for now, all there really is to know is that the alpha and the gamma secure the G protein to the plasma membrane, and the beta holds those 2 things together. So in the next video, I will discuss how G proteins become activated from a ligand coming in binding to it right here, which will then cause this to have a high affinity, and I will discuss that all in the next video.